All right, everybody, welcome to our sort of mini bioethical question of the week. This week, my question for you has to do with a new scientific technique that has been developed. Well, it's been developing for years, but it's starting to actually happen now in just the past 2 years or so. And we haven't really fully realized the potential uses for this new technology in science, but it's already raised some bioethical questions, and some people are questioning whether we should even go forward with this kind of research or whether it is not ethical and we should just stop doing it, even though we can, right? And so in science, one of the most important areas of bioethics is trying to decide what do we do about emerging technologies, new technologies. Science can only tell us what we can do, right? Engineers can only tell us what's possible. They can't tell us whether or not we should do it, whether it's the right thing to do for us. So this is where the role of a bioethicist comes in. So here's what we can do, this new thing. If you look at this image here, take just a second. I'm going to give you about 10 or 15 seconds, and I'd like you to just read some of the text and just look at the picture for just a second. Just take a minute and absorb and just kind of look at it. Whether you understand it or not, just think about what you're looking at. Okay, so if you've looked at this for a minute, you'll notice that it maybe looks kind of science-y. There's some little circles here that seem to be growing bigger, and then there's an arrow showing they get moved somewhere else, and then they're in this weird kind of tube-shaped device with a spinner, and we have some phrases up here that may mean stuff to you or not, but you've noticed that time is changing. This is the first day, and this is by day 15. What this is this is a diagram of the process of growing a miniature human brain. So in the past couple of years, biologists have developed the ability to take brain cells, so fetal brain cells, and get them to grow in a petri dish and transfer those cells to this bioreactor. And that bioreactor is able to bathe this miniature brain. It's a collection of nerves. It's shaped a little bit like a brain, but it's a little bit more blob-like. To bathe them in nutrients and feed them and keep those cells alive. So that's true. We can grow this. We can grow brain cells in culture. We can keep them alive. What is interesting to scientists is now we've gotten to the point where we can keep them alive long enough that the cells have started to begin to connect to each other and send signals, just like the brain of any other living animal. So inside of your brain cells connect to each other and then send signals back and forth. And that's how you learn and think and how you move your body and how you feel pain. It's really just a series of connections and signals being sent from cell to cell. So now we're able to grow cells that don't just live there together, but are starting to communicate with each other. These cells have never been inside of a human body. Most of them are fetal cells that probably came from fetuses that were aborted. The potential benefits of this, the reason doctors are doing this is because if we grow human brains outside of the body and we can get them to connect together in ways that are similar to actual human brains, we can study brain disorders without impacting living people, right? So these are huge benefits. I mean, we could develop new medicines, new techniques for reversing things like Alzheimer's or Parkinson's, halting seizure disorders, treating cancer, and all of that could be developed without ever actually having to test directly on a living person. And it would make our ability to learn about the brain much greater, because right now it's kind of like there's only so much you can do with a human brain, because if you poke and prod the human brain enough, you're going to damage it. Well, if you have these mini brains, they aren't anybody's brain. So there's a bit of an ethical gray area. Can we poke and prod these brains to learn about our own human brain and hopefully treat actual living humans? Are we okay with that? And here's the bioethical dilemma, right? So those are the potential benefits. Potential harms are this. As we've gotten better at growing these mini brains, those mini brains have started to show some connections that we didn't stimulate. So human researchers are stimulating most of the connections between those brain cells, but they've started noticing that these mini brains are beginning to make their own connections, which is essentially the same thing that happens in your own brain when you learn. So the question is, are these brains learning? Is it possible that they're conscious? Are they aware? Are they aware that they're a brain floating in a suspension of material? Are they able to have feelings and thoughts? We don't really know because we can't communicate with those brains the same way you and I can communicate with each other, where our brain is able to communicate with your brain through our bodies and our language. And we're not sure. Most scientists right now agree that these brains are not conscious, that there is some learning, but it's on the cellular level. But the question remains, would we know if it ever crossed that boundary where the brain was big enough and living long enough that it was starting to learn, that it was being conscious? The other kind of surprising and a little startling thing was that when they stimulated one side of the brain of these mini brains, the other side responded to that stimulus. So instead of just stimulating one cell, the other side was stimulated as well. And some of those signals look a lot like the same kinds of signals our own brains show when we feel pain. So again, the question is open. Are these brains feeling pain? Can you even feel pain if you're not conscious? We don't really know. But are we essentially growing human brains in a solution and then torturing them? We can't really tell for sure. The last thing just came out this year, actually, that I didn't even have time to add on to this, but they found that if grown long enough, those miniature brains began reproducing their own cells and growing and attaching themselves to the sides of the container and began growing a shape that looked much more like a human brain than the original cultures had. So they're starting to divide and spontaneously send signals to other parts of the brain to grow and change. All of this activity is both interesting and fascinating. There are tons of upsides to treating real human disease, huge potential benefits to being able to grow these brains in culture and test them there. But there's also some potential harms. And the question is, should we be researching the human brain by growing mini brains in solution? And I just want you to answer that question, and I'd like you to tell me, explain to me, give me your argument for why. Try to show me that you're thinking about the stakeholders involved, that you're thinking of the values and the potential bioethical approaches that you'd like to use. I don't care which stakeholders you tend to argue your position from, but you're going to need to pick a position. Okay, and you'll be posting that to a discussion, and when you've done that, you can move on to the notes activity for this week. Thanks, guys.